Good afternoon, everyone. Welcome to the 16th talk in the Only One Theme talk series Wave 2. Today's topic is Only Protection of Plant Varieties by Dr. Malathi Lakshmikumaran. Let me briefly introduce Dr. Malathi before we begin the talk. Dr. Malathi Lakshmikumaran has more than 30 years of experience in the field of biochemistry and molecular biology with an expertise in plant genomics, DNA fingerprinting, and genetic transformation. She serves as an executive director and heads the life science group at the IP division of the Lakshmikumaran and Sridharan attorneys. She is a registered patent agent and has been actively engaged in preparing, filing, and prosecuting of patent applications both in India and abroad. She mainly works on pharmaceutical, chemical, and biotechnological patent application and advances clients on plant variety protection and registration. She is actively involved in the area of biodiversity and traditional knowledge and has successfully supervised several PhD students in the area of plant molecular biology. With more than 100 publications to her credit in various national as well as international journals. And I think Dr. Premnath, you want to add it?
Well, hello, Malathi. How are you? Yeah, very happy to have Malathi today. I just wanted to point out that I'd requested Malathi to speak on this topic of protecting plant varieties. And I just wanted her to also look at it in a broader sense, look at various ways we can do it, and not just through the Plant Varieties Act, but also through patenting and so on and so forth. So, of course, she's the best person to speak about it. We're also very proud that she's an alumnus of NCL and has been an academic. So she understands also what many of us as researchers and inventors think through when we are going through the process of IP filing, commercialization, and so on and so forth. So welcome, Malathi. I think the floor is all yours. Kavita, can I hand it over to Malathi?
Yes, please do.
Yeah.
Thanks.
Okay. So thanks, Venture Center. Thanks, Mukta. Thanks, Premnath, for giving me this opportunity to talk. Always, whenever there's an NCL, then it always is special to me. It's more like an alma mater, and I was a first student of Dr. Ranjekar, but I was very, very happy to work with Dr. Jagannathan, who was my mentor. And later on, when I joined TERI, he was again my mentor. But we had peoples like Dr. Shivaraman and Rangachari and others whom I can't name. They're just stalwarts. So I feel very proud that I'm from those times. And I finished my PhD in 1979, submitted in 1980. So I'm really, really old. But again, I feel so nice. And I'm happy that I had a dual career, being a scientist and now coming into the field of patent law, where I deal with a lot of things like IP, that is patent, PVP, Biodiversity Act, and the intersection of all these, which makes it much more complex and interesting, but also challenging many times. So without much ado, let me just start. I know the theme is only protection for plant varieties, but when I had a talk with Premnath about a few weeks back, we decided that we would just expand the scope. It will be still IP with plants, but just take a little quick summary of what can get patented in plants and what the challenges we face when we try to protect something in plants as compared to something in chemicals or engineering as such. So I will quickly go and share my screen. If you can see it, let me know. And I will...
Yeah, we can see it. Can you go full screen?
Yeah, I'm also doing it at full screen. Yeah. So the theme is protection for plant varieties. And I will quickly go to my topic. So if I look at it and look at the technology that we have, the whole scenario in biotechnology did change. And what really changed is the first thing in 1980, which is a US Supreme Court decision saying that living organisms can also be patentable. Though earlier USPTO had granted some of these microorganisms, like for making cheese and also from Pasteur Institute and others. But however, we find that now there was a problem because Ananda Chakravarti went and filed a patent for a Pseudomonas, which could actually clean up the oil spills. And this actually became a huge turning point, which most of you all know that any invention under the sun using the hand of man will be a patentable subject matter. And it's not that living organisms cannot be. So anyone who wants to get into patent law, the first thing we teach them is the US Supreme Court, Diamond versus Chakravarti. And I think that opened the floodgates for a whole lot of other innovations. Then came the recombinant DNA technology by Cohen-Boyer, again, early 80s, late 70s, early 80s. And it was from Stanford University. And it saw the birth of many, many companies like Genentech, a lot of Amgens and others, a flurry of biotech companies, startups, companies came up, all because of this one Cohen-Boyer patents and their different work. And we all use recombinant DNA technology. But really, when I was doing my PhD, we didn't know what was an intron. And that's the time recombinant DNA technology. So for a person like me, we've grown up with the technology, recombinant DNA technology and all. And they collected millions of dollars as just royalties. And so if you look at growth hormone, erythropoietin, insulin, all these which are produced by recombinant methods are because of Cohen-Boyer. Oh, that was great as long as it was a eukaryote, prokaryotes. But then came the transformation of eukaryotes. It could be plants, it could be animals. Another interesting thing was Columbia University. We've also seen the rise of siRNA, and that's another example of licensing of patents. Recently, we've seen CRISPR-Cas and mRNA vaccines, which all these three, siRNA, CRISPR-Cas or mRNA, all of these have got Nobel Prizes, and very quickly after they finish their work. And so we have a huge amount of things. But today, what I will discuss is CRISPR-Cas9 with respect to plants as such. If I just do a quick summary what happens in India, I'm now only looking at plant-related innovations. You can get first when you isolate a DNA or a protein. Let's say I want to take up a gene for delayed ripening, and I want to put it into a plant, or I have a promoter which I want to put into a plant. Many times we get the problem that this can be a discovery because you're isolating sequences from nature. And these are all DNA and protein sequences occurring in nature. And therefore, so what is great about it? We also have isolated microbes, because many microbes that are there, like you have mycorrhizal strains, you have rhizobium strains, you have a whole lot of strains which can increase the yield of plants, or it can give us certain times protection against insects or other things. These microbes are not patentable because they come under discovery. So anyone working in plant-related inventions have to get out of the hurdle of your invention not falling under a discovery. That's very important. The other thing that we have a problem is 3H. Any method of agriculture or horticulture is not patentable subject matter. If I look at the old way, the way the act was written, it was actually based on asexual methods of agriculture or production of plants. It could be like rose from rose cuttings, or sugarcane from sugarcane cutting, or potato from tubers. Those type of patents could not be patentable because this is what normally a farmer would do, a breeder would do, or an agriculturalist would do. So anything which falls under asexual methods of propagation would not fall under 3H. However, our patent office is reading it much more. So if I spray any chemical or even a microorganism on a plant for it to increase yield, or let's say I put a microorganism for fixation of nitrogen. We have many of those now in the cereals, like maize. those would all come under method of agriculture and horticulture. We have some case laws, but this is a gray area. What is method of agriculture and horticulture? We don't have equivalents like that in chemical industry or in other areas like in the animal sciences or life sciences, but it is part of plant-related innovations. Therefore, recombinant microbes are patentable, but not living or microorganisms occurring in nature. You need to have modified genes to be patentable. mRNA is not patentable. cDNA is patentable. Genomic DNA is not patentable. But we can patent recombinant constructs, vectors, junction sequences. Another important thing that we find in section 3J. So anybody looking at plant-related inventions should also look at section 3J, which says plants and animals in whole or part thereof are not patentable, but microbes are patentable. But when you say microbes are patentable, see that it doesn't fall under discovery, and therefore modified and adapted, a mutated microorganism is patentable, but a naturally occurring microorganism is not patentable. Similarly, seeds, plant cells are not patentable. I have concerns about plant cell because is cell a part of a plant? My examiners and controllers will say yes, but I think the whole thing was plants and animals should not be plant cells. But today, the patent office does not give you a patent on seeds, plant cells. And that leads us to plant variety protection. Because if under the TRIPS agreement, member countries can exclude plant-related inventions, plants, and plant varieties, and also plant-related innovations, but they need to protect it under a PVP system, which is a sui generis system. So this whole thing is, but for animals, we don't have an equivalent of the PVP regime. Now, method of transformations is patentable, regeneration is patentable, method of treatment of plants should be patentable, but we have huge issues on that. But essentially, biological process for the production of plants is not patentable. So just as I said, I'm crossing A into B and producing a hybrid, or I do backcross breeding. These are things that happens in the field and are not patentable subject matter. Unrelated, you can get for nucleic acid, promoters, junction sequences, CRISPR-Cas, biofertilizers, and so on, or genetic engineering of plants, where I have many plants like the cotton, maize, oilseeds, tomato, potato, tobacco, Arabidopsis. Tobacco and Arabidopsis are more model plants. But if it is rice, wheat, maize, cotton, legumes, soya bean, all these are being transformed and genetically modified plants are being produced. And one of the most important, we all know in India, we have the BT cotton. We have Bolgard 1, then came Bolgard 2, and that's the only GM technology in India. And actually, India became a net importer to a net exporter of cotton using the Bolgard technology, Bolgard 1 and then Bolgard 2. Bolgard 2 is nothing but Bacillus thuringiensis genes which are integrated and inserted into plants. But there are other technologies also. It can be herbicide resistance. I've talked about insect tolerance. It could be fungal, virus resistance, male sterility, delayed fruit ripening, or production of secondary metabolites. Today, even production of vaccines can happen into plants. So plant is a very good system. In E. coli, you can get spills, you can have things, but in plant, you can have a controlled way of producing secondary metabolites. So let me just go, these are some of the plants, but quickly what I want to explain in this slide is that genes per se may not be patentable if it is considered as discovery, but if I modify the gene of interest. Here, I'm taking the topic of Bacillus thuringiensis or Bt toxin gene. I modify the gene because it is from Bacillus, but it has to work in plants. So I take the gene of interest and you can see a blue color thing. Into this, I modify it, and therefore the gene of interest, because it is modified and it is not what is occurring in nature, becomes patentable and does not fall under 3C, which is discovery. I then make it into a construct. As you can see my construct, it has a promoter The promoter is like an on-off switch. I also have a transit peptide because I'd like to produce that in the organelles. I have the gene of interest. Here is the Bacillus thuringiensis gene or the Bt toxin gene. And then I have a switch, which is an on-off. Now, this can be in a plasmid, which is patentable. A DNA construct is patentable. A host cell, like what I have written here, a E. coli host cell or an agrobacterium host cell, having this construct is also patentable subject matter. Now, all these are not really excluded under 3J. So till now, everything is patentable subject matter. Now I do a transformation. I take this construct and I insert into a gene. It can be by agrobacterium method. It can be by particle bombardment method. It can be by a whole lot of just trying to put it into a solution and allow the gene to get in. So there are many methods that we have, mainly agrobacterium-mediated method of transformation, and this whole construct gets inserted into the plant genome. So this method is also patentable. The construct is patentable. Now, when it gets inserted, you will see that there is a break, and this whole construct gets in. What? The promoter, the transit peptide, the gene of interest, and the terminator, which will stop the expression of the gene. The whole thing goes in. And when it goes into the plant, I have now created a new plant, which is a transform plant, which is a recombinant plant, having this gene of interest, which is expressed under the control of a promoter. The promoter can be 35S promoter. It can be any other promoter as well. But mainly, people have been using the double enhancers, which are cauliflower mosaic 35S promoters. And therefore, when it gets inserted, we have something called as a left junction, and we have something called as a right junction, which you'll see in two square brackets that I have shown here. Now, when these two junctions are there, we call these as the border sequences or the junction sequences. So the junction is part of the plant and part of the vector construct. So these junction sequences are not naturally occurring, and therefore I can claim for these junction sequences because they have been created by man. They've been created in the lab, and they are not naturally occurring. When I make it, then I do the construct. I've made my transform plant. I've got the junction sequences. I will see a plant will have only one insert, not many more inserts. And whether it is expressing, I do the assay. And then I produce the plant. And as you see in there, in my plant, I have it in a pot. And I can do the assay to show that the Bt gene is getting expressed. So till here, most of the things will get patentable, but a plant will not be patentable, even though it doesn't occur in nature, because plants and parts thereof, seeds and varieties are not patentable subject matter. So I can go the whole way, but not come to the plant. The plant is not a patentable subject matter. And here comes the problem. And this plant, which I have called BT cotton plant, I can get protection under PVP, but all other things that I've done for patenting, that is the construct preparation, doing a recombinant host cell in E. coli or agrobacterium, method of transforming a plant, method of regenerating a plant, identifying the location where it got inserted, the junction sequences, and all, or a method of identifying whether people have copied mine or not, can also be done. Only the last step, which is a plant, is not patentable. And there comes the thing that we go to the PVP. Now, because plants are not patentable, let me say I have a Bt toxin plant in the open, in the field, that's where you grow it. Anybody can copy it. All you need is some pollen. All you need is the pollen to dust it on something else, and it will get transferred to another plant. So plants are like software where I can copy it. Today, music, everybody copies it. You download and you can copy a movie, you can copy a song, you can also copy plants. But if I have a chip in my mobile phone, it cannot copy itself. You need to have a manufacturing unit, and you need to produce it. And therefore, like software, a plant can easily be copied. If it is on the field, I just have to take its pollen, dust it on my plant, and I can get it. Or better still, I wait for it to produce seeds. I take some of my seeds back home because seeds and plant varieties are not patentable subject matter. I take the seeds. I don't have to do any of these first 10 steps to produce a BT cotton. I take the seeds, I grow it, and I have BT cotton. So this is where the problem lies, and therefore the importance of plant variety protection is important. I will quickly go that today it is CRISPR-Cas. As I said, only the plant is excluded, all others are not excluded. But if I go today, it is a lot on CRISPR-Cas. We were doing the old technology, but now it is CRISPR-Cas because I can delete, I can insert, I can do a single point mutation, like we know that sickle cell anemia or any other hemophilias, you can actually do the mutation and change. But where is CRISPR and agriculture? It's great. Like I showed, we do a lot of work. But when it comes to a plant, you have to get plant variety protection, just like BT toxin and so on. So it can be virus resistance, it can be disease resistance, you can have physical and chemical resistance, you can get crop production enhancement, you can manipulate bioactive compounds and so on. I can produce plant hormones, I can produce vaccines. So plants are extremely good, but the problem that we have today is that plant per se is not patentable subject matter. And because plants can copy themselves, anyone can copy it, the protection for plants, since it is not there in the patent, it is a loose invention and people can copy it. And therefore, I'll quickly go through these. We have got disease-resistant bananas, drought-tolerant soybeans, high-yielding tomatoes, better sweet potatoes, and higher yield of rice. I'm not going to work on all these things, but just to show that these all in India can only be protected under the PVP regime. Even if you protect the construct, you do it by CRISPR-Cas, you can. But if this tomato, like you see the cherry tomatoes, once it goes out in the market, I have to just collect those cherry tomatoes and I can produce them in my garden, in my lab. But today, by CRISPR-Cas, I can actually put the capsaicin gene in tomato, or I can change the carotenoid content in tomatoes, and therefore I can get a spicy tomato as well, or a higher rich carotenoid contents as well. So these are some of the examples. Corteva is giving the license for research for CRISPR-Cas. There are more than 13 genes that have been put in rice. There is the Xanthomonas oryzae, which is a zoo strain for bacterial blight that have been also manipulated to get you disease-resistant rice strains and so on. We can also get a black rice, which is anthocyanin production in rice, where I can actually put carotenoids and you can get a black rice as well using CRISPR-Cas. I can also have this calyx soybean, where I produce high amount of oleic acid. Now, oleic acid is important because the higher the oleic acid, more stable is the oil. And therefore, today, if you look at brassica mustard oil, has a higher amount, but not as much. So people would like to have a more stable oil, and so it doesn't get rancid, and you need to increase the high oleic content. And that can be done by CRISPR-Cas. And this is known as Calino oil. It is very healthy oil, higher oleic acid, lower other acids like linolenic and others. And this type of oils can be manipulated into olive, into sunflower, into safflower, into mustard, and so on. So it's very, very easy to do CRISPR-Cas mutations in a gene called GMFT2A, and can also be used to delay the flowering in soybean, to change the oleic acid content. And so we have all these. I won't go into this. You can also gene edit a banana to have a more nutritionally important banana, or a delayed ripening in banana. It is being done by Nabi in Mohali as well. What I want to tell here, we've looked at it, it's great, patents are required, but is patent the right way to go ahead to protect your varieties? Because plants, seeds, varieties are not being patentable in India. And the only way that you want to get a full solid protection is through the PVP regime. So let me just talk about antibodies for a minute. I produce antibodies in E. coli, or I can produce them in CHO cell, or I can produce antibody in a xenomouse. Okay? But these xenomouse, you can actually control them in the lab. The cell lines can be controlled in the lab and they don't go out in the market. So if somebody wants to copy another antibody, they have to legally obtain the cell lines, or they have to legally obtain the xenomouse, having the gene of interest in that. And therefore, even if you look at antibodies, or I want to produce insulin, it is not easy. You need to create that insulin gene in E. coli, in a fermenter and produce. But in plants, let's say I have a plant which can produce insulin. It's so easy. I take the seeds and I can cultivate it, and there it goes. So PVP is required if you want to get a protection for plants, and the patent may not be the full regime, because if you catch a farmer and say, you're copying mine, he'll say, I've only taken your plant variety. I have not done any of the recombinant constructs. I have not used CRISPR-Cas. I have not selected for the plants and so on. Because India does not allow protection for plants, which is there. I just want to go before that. If I look at US, we have something called as plant patents. Plant patents are those patents, came in 1930. A variety can be completely protected as one patent, as a patent, giving all its characteristics, giving all its traits, maybe agronomic characters, phenotypic characters. Of course, we don't give genotypic characters, we go phenotypic characters. In US, you can protect a variety under a plant patent, unless they are asexually produced plants like potatoes, you cannot, because these cannot. But any others which are produced through seeds, you can protect it, something called as plant patents. Second, in the US and Europe, I can protect a plant. In Europe, I cannot protect a plant variety, like a specific BT cotton variety, I cannot. But plant, a BT cotton plant, in any variety it is. So if I'm the first one to produce a BT cotton, a BT gene into a plant variety, I can claim for the whole plant. Not as a variety, but as a plant in Europe. In US, for the plants per se, I can get a patent protection. So if I look at my earlier scheme, right from the construct to the method and finally the plants, I'm not talking of plant variety, I'm talking a plant possessing high oleic acid using these constructs, a plant having BT toxin, a cotton plant having BT toxin, a soybean plant having high oleic acid, a rice plant having resistance to xanthomonas. All these as a broad category can be protected in US and Europe and many other countries, but not India. India, because you want to protect the farmers, plant and plant varieties, seeds are not patentable subject matter. So here, if you people do come out with inventions, India, you may not be able to do it. You'll only have to go through a PVP regime, but you can do that in other countries. Because the farmers and whoever is going to copy it, not going to use your construct, are not going to do CRISPR-Cas technology. All they're going to do is to take your seeds and grow it in their field. So what is PPVFRA? It is a protection of plant varieties and farmers' rights. So we have two rights here, the breeders' rights and the farmers' rights. Now, even though this act came up in 2001, is because the TRIPS mandated India that you need to protect your plant varieties either through a sui generis system or through a UPOV mechanism, and you can get it protected. But plant varieties have to be protected either through patents or through a PVP system or both. So if you look at US and EP, There are plant varieties in US. You can get plant variety as a plant patent. You can also get it as a utility patent, and you can also get PVP. So in US, you can actually get double protection. I can get a patent for, say, BT cotton, and for a specific variety, I can get a PVP for it. In Europe, I can get a plant, a utility patent, a plant having BT gene, expressing BT gene, or expressing a gene to increase folic acid is also patentable in Europe, and I can get a PVP variety. So varieties are not patentable in Europe, but in US, varieties are also patentable under the plant patent, okay? So since we don't have it, most of the people working on plant varieties need to take protection under the PPV FRA. So for short, I'll call it as plant variety protection. And this is what you have. India is unique because we are not members of UPOV. UPOV, there was 1978 and 1991. We did not become part of it. We are now have something called as PVP, which is a protection of a variety. You can get protection for a new variety, which is not in public domain. You can do a trial for 1 year, but within 1 year from your commercialization, you need to protect it. Then it falls under new variety. If it is something outside India, then it should not have been sold more than 4 years. So if a new variety of potato or a new variety of cotton, that should not have been sold outside of India for more than 4 years. Then it comes under new variety protection. So in a variety protection, what we do is all the characteristics are studied, and I get a protection for one variety. All its phenotypic characters are put in. I will say what is flower color, what is its height, whether it's disease resistant, what is its yield. But it's just that variety that is there and nothing more that I can get for it. So India is unique. Every country has new variety protection, but India is unique because we have something called as the extent variety protection, which is something which is already into public domain. The reason is we were very slow. Because we came in late, any variety can get protected under extent variety, or if it is notified under the Seeds Act, or it's a farmer's variety, or any variety which is in public domain and you've read it, you can go and file protection under extent variety. A farmer's variety may not be having that uniformity, but it could be land races or any variety that a farmer has, and that can get protected under farmer's variety. The extent and new varieties goes under breeder's variety. Then there's essentially derived varieties, and I'll talk about that very quickly. So we have about 189 species. It may be now 192 or something. I'll check that out. Species which are governed, which are under the registration process. There's something called at the Seeds Act. So you can go and notify certain variety under the Seeds Act. That can also get protected under the extent varieties, similar to the farmer's variety. Now, what do I mean by farmer's variety? I'm sure you'll ask a question. The farmer can cultivate his own crops. He can do it either by himself or by somebody else. So if you are a big politician, let me say, and you've given it to somebody else to cultivate it, you politician can also be considered as a farmer, because he is either you cultivate the land by yourself or you cultivate by supervising the cultivation of land through any other person. So you're a businessman, or you are a politician, or you're anybody else. You own a land, you don't directly be a farmer, but you supervise that land, is also considered as a farmer, and his variety will be considered as farmer's variety. Anybody who conserves and preserves varieties will also come under the farmer's thing. So a farmer's variety is something which is traditionally cultivated, evolved by farmers, because like in chemistry, I can create a molecule in the lab, but I cannot create a new variety in the lab. I have to take some variety, make changes either through backcross breeding or through F1 breeding, or make a hybrid, and only then. So all varieties have come from some other variety, and you need to say how it is made changes in. I'll just take a minute on essentially derived variety. And for that, I just want to give an example. Just a minute, I'm going to show an example of what is an essentially derived variety. Let's say I have a cotton variety called BCH. That is how we give all the characteristics. So if it is a cotton, we have more than 37, 38 characteristics that you have to give. What is a hypocotyl pigmentation? What is a leaf to petiole pigmentation? What is the plant height? What is the color of the petal? What is the color of the pollen? What is it? How does it open? Early flowering, late flowering. In rice, is it scented rice? Is it basmati rice? And so on. And so what you do is, when I have a variety called VCH1. Now, when I have a VCH1, I produce a VCH1 with Bolgar2. That means what I did, I took VCH1 and I got, by backcross breeding, I added Bolgar2. So what happened is, there was this variety called Cocker in which you put your Bolgar2. That is two genes, Cry1AC and Cry2AB. Now, because you have to have it in the same location, I will take my Cocker variety and I'll backcross it. I'll cross it with VCH1 and I'll keep backcrossing with VCH1 so that I get a near isogenic line of VCH1. That means it's very much like VCH1, but it has Bolgar2. How do I do it? I'll do it through DNA testing. Every time I cross, I do a backcross. So first I'll cross VCH1 and Cocker. Then I'll keep backcrossing with VCH1 so that I end up with a variety which will have Bolgar2, but will look exactly like VCH1. And we call these as near isogenic lines. So this near isogenic line that I created is actually considered to be an EDV. That is, it has essentially all the characteristics of VCH1, but it has a Bolgar2. That can be Bolgar1, or it can have Bolgar2. Or I say I've changed the oleic acid, then I will say it is only oleic acid gets changed. So if I look at the characteristics, VCH1 and VCH1 Bolgar2, I can't see any change. Why? The hypocotyl pigmentation is the same. The leaf to petiole pigmentation is the same. The plant height is the same. The flower color, petal cover is cream in both. A ball, that is the prominence of the tip, is it rounded? Is it pointed? Is it blunt? Even that is the same. The weight of seeds per cotton ball is also the same. You've got large number of seeds per cotton ball. But what is the difference? It is a transgenic trait called BT trait, which is a Bolgar2 gene. So Bolgar2 has two genes, Cry1AC and Cry2AB. In the VCH1, I do not have the Bolgar genes or the BT genes. Here, the only difference is the Bolgar genes or the two genes that I have. So if I look at to the eye, I cannot find out any difference between VCH1 and VCH1 Bolgar2. Therefore, VCH1 Bolgar2 is very much like VCH1, but different only in the characteristics called Bolgar or BT toxin. It can be one gene or two genes, and I can only test it using PCR or an assay method, but it will be. So this is what we call as what I call that VCH1 Bolgar2 is an essentially derived variety of VCH1. And I've done it clearly only through backcross breeding. Okay. But if I compare a variety X, that also has Bolgar, if you see the trait 38. But if I compare the other variety, other characteristics, the hypocotyl pigmentation may be the same, but the leaf to the petiole pigmentation is absent in variety. The plant height is tall, whereas this is medium. The flower color is yellow, but here it is cream. The ball tip is pointed, whether it is. The number of seeds per cotton ball is medium here, whereas others is large. Therefore, variety X is different from even VCH1 Bolgar. The VCH Bolgar, both of them have BT cotton in it, BT genes in it, but because they are different, variety X cannot be considered as a essentially derived variety of VCH1 Bolgar2. Why? Because the other characteristics are different. It may be same as respect to BT, with both Cry1AC and Cry2AB because I did the same backwards beading. But variety X is a different variety having ball guard, but it will get a completely different denomination and it can get protection. And that is how, if I have a BT cotton, for each of the varieties having ball guard 2, I need to go and take protection. Because if anybody says you're copying mine, the PVP office will compare all the traits. There are some 37 traits. All the traits will be compared. If there's even 1 or 2 trait differences, then it is considered a different variety. So it is that narrow. Just like in US or other countries, plant variety protection is for a protection of your phenotypic characters, what I can see. It is genotypic, but we don't check it. But seeing from the eye, looking at the agronomic characters, looking at the phenotypic characters, I will say. So all 3 are different varieties, but VCH1 ball guard 2 is essentially derived from VCH1, and variety X is not derived from these, and I can get protection under either new category or extend category, depending when I sold it. If I look at what is a variety, what is a variety? In our whole act, it defines what is a variety. And this is very important for me. A variety is a plant grouping. It's a plant grouping. A variety will be a plant, but not a single plant. It's a plant grouping. So if I have Pusa Basmati as a plant, I'll give the characteristics, but I'll give it as a group. All of them are Pusa Basmati. I take 10 of these, put them together, grow it together, and show it as a Pusa Basmati. Pusa Basmati is a plant. That means I can get variety for a rice. And rice is Oryza sativa. It can be indica, it can be japonica. But wild varieties of rice, we are not giving protection for. So a variety is a plant grouping. When I say plant grouping, it is called Oryza sativa. It can be indica, it can be japonica, but we are only allowed for plants groups which falls under Oryza sativa. But any other species of rice, there is raffinose, there's so many others, they will not fall under that grouping. We don't have microorganisms are not covered under PPVFRE. The plant grouping should be a single botanical taxon of the lowest known rank. That is, I just cannot say Oryza, because Oryza is a species, it's a genus, and it covers many species, correct? So Oryza is the genus, and Oryza sativa is the species. And this is the lowest rank. I can't go below that, because all the varieties of Oryza sativa will fall within this rank called Oryza sativa. Okay, so this is how you have to understand and say, I can get protection for any varieties or strains of Oryza sativa. Now, this, it should have the expression of the characteristic. Now, even though Bt toxin through my eye, I cannot find out, but if I put insects and the insects don't eat the plant, that means it is resistant to insect. Why? Because it is expressing the Bt gene. So then how do I differentiate the varieties? It should express certain characteristics resulting in a given genotype of the plant grouping. And it should be distinguished from any other plant grouping by expression of at least one characteristic. So I said cotton will have, let's say, 37. Rice may have about 65 characteristics that is given by the government of India under a DUS guideline. And therefore, when I look at those 37 characteristics for cotton, even if I can show one color, let me say petal color is generally cream. I have come up with a petal color which is blue in color, for instance, or even mine is dark yellow as compared to cream. I can get a variety protection just for that one characteristic. People will say, what's the use of having a flower color? What is it? But that itself is enough for me to get a PVP protection. I just need to show one characteristic which is different. And that characteristic should be governed by an expression of at least one gene and you can't tell what the gene is, but you will see based on the phenotypic characters. Now, it should be considered as a unit, this variety, which is with regard to suitability for being propagated. It should be able to propagate. So if you have GERT or something which doesn't propagate, you'll have a problem. And it should remain unchanged after such propagation. Year after year, if I propagate, it should be, if it is Pusa Basmati, it must remain to be Pusa Basmati year after year. If you cross it with something, it will change. But I need to keep a field, or I need to grow it in a way that there is no crossing and that Pusa Basmati remains as Pusa Basmati. So as I told you, a variety must be a grouping like Oryza sativa. It must have at least one chain, one characteristic should be different from all other plants. Third, it should be able to propagate. That is, I need to give seeds, or in potato, I need to give tubers, such that it will remain year after year, it remains the same propagated material. Now, the propagating material of a variety can be a new variety, can be an extant variety, it can be a transgenic variety, it can be a farmer's variety, and or an essentially derived variety. And therefore, if I give this thing, these are varieties. I've only given certain characteristics, but these are varieties. You need to give all the 37. If you're going to cotton, you need to give all the characteristics that you have looked at in the field for 2 years at 2 different locations, because due to environmental conditions, we can have G into E and you can get changes. So if I grow mustard in Delhi and I grow mustard in Leh, the characteristics will be different because of the environmental condition. But the genotype remains the same. So I'll give the characteristics of what the characteristic is in Delhi region or in the North India region. And this is how I get my protection of my plant varieties. You'll have any questions, or should I continue?
Malti, there are some questions, but if you wanted to finish a few
Yeah, I
slides
think
and
I
then we
should
can close
stop
after
here.
that. Okay.
Yeah.
Okay.
Should I stop sharing and
No,
take
no,
questions?
you can keep it in case you want to address some
Okay, fine.
questions using some slides,
Yeah,
right?
correct.
I would, by the way, there are some questions that are coming up in the chat box. People are welcome to type it in. But at the same time, if somebody wants to ask their questions, they can use the reactions button and press the raise hands, and we will ask people to take it up as well. I just wanted to start by asking if you could take up a couple of examples, like you
Sure.
mentioned some of these CRISPR edited plants, right? And illustrate with what kind of claims have been used to protect them, right? You know, what that's one type of an example. Another kind of an example is take a specific plant and say between US, Europe, and India, you know, what were the kind of different claims and patents or PVP protections that people got?
Yeah. So I'll just explain this. If I go for a variety, now let me tell you, in the US, when you go for a plant patent, you're supposed to give inside all the characteristics. Suppose it is a cherry tomato. I showed you cherry tomato, correct?
Yeah.
Versus a regular tomato. What am I claiming for? I'm claiming for a cherry tomato. Okay. Let me just quickly show that cherry tomato. Let me give you that example. Or I'll just give you an example. Let me give you this example to show what I mean. Here I have a cherry tomato. Now the thing that they have created is they've created a cherry tomato. Forget it's by CRISPR-Cas or whatever it is. It can be by any method that you created. So when I claim for cherry tomato, my claim is not only for the tomato. The claim is for the whole plant, the plant with its roots, with its height of the plant, and tomato, all the characteristics of the plant have to be given. You can miss one or two, but you need to say what is the pollen color. You need to say whether it's early flowering or late flowering. How much is the seed weight of these cherry tomatoes? Per plant, how much yield will I get? So when I say I got a claim for a PVP for the cherry tomatoes, It is for the whole plant. Okay? It is not only, my claim is not for the cherry tomato. It is not. It is for the plant height, the pollen color, the early flowering, what color of the fruit, and everything. So I have more than 30 characteristics of tomato. I need to give all the characteristics and I need to claim it. However, there may be only one difference. So let me say the second tomato versus this. I got every tomato is tomato. So most of the characteristics will remain the same. But one, I can say it's a bushy tomato. Because you don't get tomatoes like this. When you get a cherry tomato, you'll get it like a long chain with tomatoes hanging out. Like you'll get a chain. But mine is a bushy tomato. So therefore, my difference from the cherry tomatoes are they come in a long chain. Mine comes as a bush. But when I claim when I want to say infringement, I don't go based on the infringement that it is cherry tomato versus another cherry tomato. I have to say your plant, either the genotype, I will take the sequence data of the whole genome, or I have to grow this cherry tomato from beginning till end. I must show all the characteristics are matching. So it is like identical twins. Only identical twins are similar in the genome, correct? No two siblings are same, but an identical twin will be similar. And when I have to say your identical twin, you may look same, but one might have a short hair, one may have a long hair, but genotypically you are the same. So that is where you must remember. You must not say, oh, I'm bushy tomato, I'm cherry tomato, I'm red tomato, I'm orange tomato. No. A PVP is given for the whole plant. It is not given only for a specific characteristic. The difference from cherry tomato, which is in a string, versus a bushy cherry tomato, may be this. But when I ask for a PVP variety and I want to show somebody is infringing on it, all the characteristics must match. And therefore, it is important that I give all the characteristics. So let me look at in the US plant patent, I give all the characteristics. If there are 30 characteristics for tomato, I give all the 30 characteristics, and all the 30 characteristics must match. You just cannot say yours is bushy tomato and cherry tomato, bushy cherry tomato, and therefore mine and yours are the same. They will not accept. Therefore, in PVP, it's very strict. All the 30 characteristics that you've given must match, and also the one that was different must also match. Okay, so this is where in a plant variety, the variety is for its genetics, it's for the whole genetics, but we don't go and do the sequencing. We only see the expression of the genes, and therefore we give for the phenotypic characteristics. Many times when people want to show that you have copied mine, we will take my variety, we will take the competitor's variety, and we'll sequence whole genome sequencing, because it has become very easy now. And then I have to show it exactly matches. Even if there are few differences, I will have a problem. And therefore, when it's a hybrid, I have to show parent A, parent B are same. When I cross it, I get this hybrid, and this hybrid should have this characteristics. Okay. And if I look at this tomato down, which is having red fetter, look at all these. These are just variation in the color. The plant may look very much identical, but the minute I get variation in the flower color, or I get variation in the pericarp, or I get variation in the pulp, each of it will get a variety designation. Okay, so that is where the difference is. A PVP is for the whole characteristics of the plant. It is for the genotype of the plant. But a genotype cannot be given. Therefore, we give the phenotype. Okay, so that is where it is. Even one BT gene, or I change the oleic acid content by modifying one gene, it is a different variety altogether. All 37 characteristics of cotton might match, but there's one gene which will not match. Okay, and that is how I get it. So remember, for plant variety, if you want to litigate, it has to be identical, like an identical twin.
Yep. can you also, for example, if you take a gene-edited tomato plant, can
Yeah,
you
like
tell
this.
me in the US, what kind of claims would be there in it?
Okay, so this is good. So in US, you can get double protection, I said, correct?
Yeah.
One, I will get, I will tell you what I have done. I will get under the plant pattern, let's take this variety cherry tomato, correct?
Mhm.
In cherry tomato, I have to show what gene have I manipulated. So I will give the target gene and I will show the exact manipulation, how I manipulated. There are three genes called the PSY. There is one MYR and there's a GR, something else. There are three genes. So let's take the PSY, which is the phytoin synthase gene.
Yeah.
Yeah.
Sorry, were you looking for something?
No, I was thinking whether I have enough power in my, yeah, it is there. Okay. So my question is, in my utility patent, I will only claim for that deletion I created or an insertion of a gene. So there are three genes. Either I can insert all the three genes, the red tomato will become green. And then I can do backcross breeding. I can take two genes, I can take single gene, or I can take three genes, or I can take any two. So the minute I do that, I'm only claiming for a deletion or insertion of one gene. And I'll show how the deletion took place and how the insertion took place. So a tomato with an insertion at this insertion. It doesn't matter where in the gene, where in the genome is. So if I do a CRISPR-Cas, I'll just say insertion of a gene and this, or deletion, I'll show the exact deletion and so on. I'll also claim for the vector, which has the PAM, which has Cas, which has the complementary sequence as well, which we give in the guide RNA. I can claim just for the vector with the guide RNA, having PAM and Cas9. That's enough. Because anybody copying my vector will also be infringing. Then I will go and claim for the deletion or insertion of one of these sequences, which you can see here. They have edited different types of genes here, and therefore you get the different colors. So that's how I will do it in the utility patent. But in a plant patent, just like our PVP, I need to give all the characteristics of the plant, including its fruit, including the pollen color, including the height, and so on. That is under the plant patent. Under the utility patent, I will have for the guide RNA, I'll have the vector with the guide RNA, and then I'll have a host cell with the guide RNA, and then I'll show the deletions or insertions that have taken place, and then I'll claim a tomato having this insertion, or a tomato having deletion in sequence ID numbers so and so, or a tomato having deletions or insertions by a PSY, phytoin synthase gene. And I can give the sequence of the phytoin synthase gene or knockout of the phytoin synthase gene. Those are claims in the patent. In India, I can claim for the guide RNA. I can claim for a vector with the guide RNA. I can claim for the method of making a tomato with this guide RNA CRISPR-Cas, and it stops. I cannot get a plant claim, correct?
Yeah.
So just like I showed you before, I can get for the vector, I can get for the guide RNA, I can get for the host cell, I can get for the method of making a tomato using CRISPR-Cas. Done. With that, you stop. Now, if a person is going to copy this cherry tomato, he doesn't have to do CRISPR-Cas. He has to just buy this in the market, put it in his field, and produce it, correct?
Yeah.
Why does he have to do CRISPR-Cas? And my claims are for CRISPR-Cas, my claims are for deletions. But in the US, a plant cell having this deletion is enough. In India, even if they gave me a plant cell with this deletion, it is enough. And I will say Solanaceae. I will not even say tomato, because Solanaceae is the genus.
Yeah.
And any of the Solanaceae will have this gene, correct? So when I want to go broad, I will go. But in a plant variety, it has to be tomato. It has to be Lycosporium esculentum. That's it. It has to be. So in US, I can catch anyone using CRISPR-Cas. I can use anybody with a product having my CRISPR-Cas. In India, I cannot catch a farmer saying that you have got CRISPR-Cas of mine. I have made these cherry tomatoes using CRISPR-Cas, but you cannot catch a farmer. Because farmer will say, sorry, I didn't do it. I took a copy of yours and I used it. You don't have a plant claim. Be done with it. But I will catch the farmer if I have a PVP variety on this. Okay?
Yeah.
Now, if he's a smart competitor of mine, he will try to cross it with some other variety such that it is not an identical twin, correct? It's not identical. He'll take these cherry tomatoes. Farmer may not do it, but a competitor of mine will do it, copy something else, put something else, and therefore he can copy. Therefore, if you look at BT gene, there are hundreds of people selling BT gene, that is BT cotton. None of them have done the research. All they have done is back crossbreeding. Or just take your competitors, put it in the soil, produce the seeds. You don't have to do that. You have to give it to the contract farmer. He'll make the seeds and give you.
Yep.
I hope I've answered your question.
Yeah, I think that's quite clear. I wanted to just, I don't know how much time you have, maybe 5 more minutes if you don't mind.
5 more minutes
max,
I can take, yeah.
Max, yeah, maybe 2-3 minutes would also be fine.
And we can always take another day a call with a lot of questions.
Sure. There's also a question on, whether in order to get a variety protection, people need to just put down all the phenotypic characteristics, or they should also be looking at the genotypic characterization of some of their plants.
No, we don't allow, because today PVP authorities are not taking genotypic. You can make and keep it in your record, but today there is no genotypic. It's all phenotypic markers. Except when you have BT cotton or you have high oleic acid, or let's say I have put a capsaicin gene into a plant, I can give these extra tests and I tell them that you please test for this. Okay? And if you feel somebody else is copying mine, I can test it. But let's say anybody infringing a cherry tomato has to identical to that plant. The minute they cross it with something else and make a small variation, they'll get another plant variety, and I can't go after them.
There is one other question about Section 39, statutory provisions. Section 39 of the Indian Patent Act also mandatory for plant protection foreign filings. That's a question which has come from Niranjan.
Section 39 under the Patent Act?
Statutory provisions, Section 39 of the Indian Patents Act. Niranjan, would you like to take it on your verbally? I will need to explain that if you can. Just give me a minute.
No, Section 39 of the Patents Act.
Yeah.
Go ahead. I didn't.
Niranjan?
Yeah. Am I audible now?
Yeah, please go ahead.
Yeah. Because you're talking about patent filing, foreign filing.
Yes.
Yeah, just maybe Niranjan, you can take a minute to explain your question.
Yeah. I know he's talking. 39 is foreign filing.
Yeah, FFL. Yes. So is it necessary to have FFL? Yes. If you go, let's say for example,
FFL is banned for anything which is under the terrorist or defense or atomic energy, or you have something which is dual use technology. So if I have come out with a new variety of tomato, I can either file in India, I have to obtain foreign filing license only if there's an Indian resident inventor under Section 39 of the Act, Patents Act, okay? Though India will not grant FFL if it is some terrorist or defense related or nuclear related. If it is to make these cherry tomatoes, they won't stop you. But you have to take permission before you go and file outside of India. So let's say India does not allow utility patents for this cherry tomato or tomato with capsaicin. I've come up with this very good thing. India will not allow a patent. So you'll say, I don't want to file a patent in India. You'll go to Section 39, file for a foreign filing license, and then go file either PCT or a US application. Niranjan, have I answered your question or not?
So we need to take a permission, that's it.
Yeah. You need to take a permission and it takes 2 weeks if you want to file outside of India. For these type of inventions, you will not face a problem. But anything which is defense related, or dual use technology, you will face a problem.
Yeah, so it's
Yeah.
like waiting for almost like 21 days, even if you are to apply for any.
Yeah.
Yeah. So what we
Ma-
do is, when it is a very invention is being drafted, we write a 2-page thing and submit it. By the time you draft your whole invention, you'll get your FFM.
Okay.
You don't need the complete invention.
Yeah. Thank you.
Malti, if I'm filing in the US eventually, should I be filing a provisional in India to get my priority date, even though I know it will not get approved in India?
Yeah, so there are two things. Go to section 39. One, I can file in India first, a provisional, wait for 6 weeks, and then file in US or PCT. Okay, that is one route. The second route is I don't file in India. I take a foreign filing license and I file directly outside. But for most Indian companies, Indian institutes, we do file a provisional application in India, because a lot of people also file for software which doesn't get allowed in India, or things like this where I want to put a CRISPR-Cas, correct? So India will not give the plant claim, but it will give everything else. So if to be on the safer side for CSIR and others, we file a patent application in India, wait for 6 weeks before I take it outside of India. Because in the 6 weeks, the patent office is to look at it and see whether it comes under the dual use technology or not. And therefore, and you can take priority from India, even though India may not grant such patents.
Yeah, thank you. So I just wanted to let people know that there's a feedback form which you can fill up, which is on the chat box. I wanted to just ask you just two quick questions. One, if there are people filing PVPs in India, do you want to give them few pointers on what's the best practice and what they should do and what they should get to? And then finally, there's
so
one
I
last
would
question
ask
which I'll
them to
take up
go,
here.
first they should go onto the PVP, stop getting it. First, they need to go on the PVP website, PPVFRA website. On the website, if you're going to do for tomato, there is the dust guidelines. When you go to the dust guidelines, you have to fill up that form based on the dust guidelines. So you will take 1 to 2 years first to get all the 30 characteristics of tomato. In 2 seasons you need to get. You need to have something which is very similar to that and give all the details and then apply. It is not like that I did half my work and I did. And you also need to show that it doesn't segregate. Because if it segregates during propagation, you have a problem. So first thing that we tell people is, if it is tomato, it is cotton, if it is mint, anything, you go and look at the dust guidelines. Every of those dust guidelines, you need to see what your characteristics are. And you need to do it for 2 years and in 2 different locations if possible. Even if you don't do it in 2 locations, you need, because you need to get what the different, and then you fill up form 1 based on these characteristics, and you have to say, my variety is different from others as compared to this. And you're supposed to give 2 reference varieties which you think are close, and you need the dust characteristics of those reference varieties as well. So yours plus 2 more, you need to get all the characteristics. If you give only 20 out of 30, it is going to get rejected. So plant breeders do this day in and day out, and they have all the characteristics ready with them.
Yep.
You cannot think about it and do, you have to actually go to the ground and do it.
Yeah.
So it takes 2 to 3 years for a plant variety application to be made.
Yeah. So my last question, I know there will be more coming, and there was a hand raised also, but I think we have short of time. Do you want to give a little bit of a short sort of example of, somebody has mentioned this PepsiCo India losing the PVP certificate in potatoes in 2021.
If people can wait, I will just quickly go through that PepsiCo case as well.
Yeah.
Just 2 minutes, and
Sure.
can you
Thank you.
see?
Yeah, we
you
can see your
my
slides.
screen?
Yeah. Yeah.
Yeah, I did it. I know people will ask. I'm sorry, this should be like a 2-hour program. I've not done I've not done justice to this work today.
Yeah. I think we'll have a longer session with you,
So this is
Malti.
what happened in the PepsiCo case. So this was a breeder who created this, you have these potatoes which are very, very long. So when you make, you get those long French fries, correct? If you look at each French fry, it's very long. So you need a potato which is that long. And there should be a certain taste and so on. So what PepsiCo India did, they filed the application in their name, PepsiCo India holding name, and filed for a new variety. And by 2016, they got it granted under extent category because this variety was well known in the US. So it could not come under new category. What then happened is Pepsi introduced this variety in 2009. It's a contract farming arrangement with 12,000 farmers, and we'll buy back everything. But a lot of the farmers also kept some things back. And as you know, with potato, you keep it in your house, you'll get tubers. You take the tubers, you can grow another potato from it. It's just like in plants, you can copy any plant. And therefore, some of the farmers started saying, why give it back to PepsiCo? We can give it to our friends who can also make the French fries or potato chips and so on. But then PepsiCo sued 9 farmers and said that you are copying my variety. So variety has to be identical variety, as I told you, all characteristics to match. But because they went and sued 9 farmers, this was a backlash for PepsiCo, and they withdrew the lawsuits and said, okay, we are not fighting against farmers, because even under Section 35 and 39 of the PPVFRA, it's difficult to sue farmers because it's very, very political. But there was one activist called Kavita Kuruganti who said, no, I think this potato variety has to be revoked. And she said, one, there are a lot of issues with this potato. And she said, the one reason why it got revoked is this. PepsiCo India Holdings filed the patent, and the breeder was Dr. Hoops. Just like in patent, you need to assign your rights. Dr. Hoops assigned its rights to Frito-Lay America. Frito-Lay America should have then assigned its rights to PepsiCo India Holding. They're all same company, they're subsidiaries, but their entities are different. Hoops gave it to Frito-Lay. Frito-Lay did not in turn assign its rights to PIH, which is PepsiCo India Holding. But who filed the application? PepsiCo India Holding. Now the patent got granted because they went and told the registrar, we are the same company. Why are you making such an issue, this, that, and everything? Later on, they said, yes, we do have an assignment. And the assignment is between Frito-Lay and PIH. But that is an oral assignment, okay? Now, they should have come back because oral assignment, even in Patents Act, if there is no assignment, the patent will get lost. Broad Institute lost its patent just because the assignment was not there. The PPVFRA said, I'm revoking this patent because PepsiCo India Holding did not have the rights from Dr. Hoop, who had given it to Frito-Lay. Frito-Lay should have given the assignment to PIH, and PIH should have filed this application. In other words, if Frito-Lay North America had filed this application, because they had rights from Dr. Hoops to file it, no problem would have happened. Because they said it was an oral assignment between entities, the chairperson then revoked the patent, revoked the PVP registration for this. It went to the courts, the single judge said, yes, assignment not there, I think it is correct. It went to the division bench in Delhi High Court, and they are looking into it, and they said, look. Now, PIH could not have filed it. There would have been an oral assignment. You, as a PPVFRA, you granted it. You, as a granting authority, now cannot say, I made a mistake. I thought everything was correct. And even my mistake, I am revoking this patent. So right now, it's with the division bench. And but right now, it's all revoked. There is no stay. And the only reason, there's nothing wrong in it. Therefore, it is very important that when you file it, You cannot give incorrect information, and your assignments have to be correct. In this, the only reason why it got killed was there was no assignment from Frito-Lay to PIH.
Very interesting. Thank you very much, Malathi. I think I've taken a lot of your time today. We do hope to have a full session learning from you. I think you're the best teacher for IP that I've ever seen. So great.
No. I think that's I'm a best teacher, but I don't think I'm a best teacher for IP.
Okay, so with this, I think we will close. Thank you very much. I'll hand it over back to Kavita, and Kavita, please, you can close the session today.
Gentle reminder to everyone to fill in the feedback. The link is shared in the chat box. Our next talk is on 14th of May, again by Dr. Malathi Lakshmikumaran, and the topic is cross-border collaboration in biodiversity. So do join us on 14th of May.
Yeah.
Thank you.
Thank you very much. Thanks a lot.
Okay. Thank you. Bye-bye.
Bye.
Bye-bye.
Thanks, Premnath. Thanks, everyone.